Microglia are a type of neuroglia, glial cell, located throughout the brain and spinal cord. Microglia account for 10 to 15% of all cells found within the brain. As the resident macrophage cells, they act as the first and main form of active immune defense in the central nervous system, CNS. Microglia, and other neuroglia including astrocytes, are distributed in large non-overlapping regions throughout the CNS. Microglia are key cells in overall brain maintenance. They are constantly scavenging the CNS for plaques, damaged or unnecessary neurons and synapses, and infectious agents. Since these processes must be efficient to prevent potentially fatal damage, microglia are extremely sensitive to even small pathological changes in the CNS. This sensitivity is achieved in part by the presence of unique potassium channels that respond to even small changes in extracellular potassium. Microglia are the permanent resident immune cells of the brain and spinal cord, sharing many similarities with macrophages, the cells that destroy pathogens, outside the central nervous system. First impressions were underwhelming. In the 1800s, the pathologist Rudolf Virchow noted the presence of small round cells packing the spaces between neurons and named them nervenkit, or neuroglia, which can be translated to putty or glue. One variety of these cells, known as astrocytes, was defined in 1893. Microglia themselves were first identified and characterized by Spanish neuroanatomists Nicolas Achucarro and Pio del Rio Ortega, both students of Santiago Ramón y Cajal, the undisputed father of neuroscience, early in the 20th century. Urban legend has it that Del Rio Ortega suggested that microglia looked like aliens from another realm, which is, metaphorically, not far off given their origin in the fetal yolk sac rather than the neural ectoderm from which all other brain cells develop. The relatively late entry of microglia into the neuroscience field a century ago may be in part responsible for the limited attention and understanding they have received. But since their origin was fully described 7 years ago, the importance of microglia has gradually been recognized. The ability to view and characterize different neural cells, including microglia, began in 1880 when Nissl staining was developed by Franz Nissl. Franz Nissl and F. Robertson first described microglial cells during their histology experiments. The cell staining techniques in the 1880s showed that microglia are related to macrophages. The activation of microglia and formation of ramified microglial clusters was first noted by Victor Babes while studying a rabies case in 1897. Babes noted the cells were found in a variety of viral brain infections but did not know what the clusters of microglia he saw. Rio Ortega, a student of Santiago Ramón y Cajal, first called the cells microglia around 1920. He went on to characterize microglial response to brain lesions in 1927 and note that fountains of microglia present in the corpus callosum and other perinatal white matter areas in 1932. After many years of research, Rio Ortega became generally considered as the father of microglia. For a long period of time little improvement was made in our knowledge of microglia. Then, in 1988, Hickey and Kimura showed that perovascular microglial cells are bone marrow derived, and express high levels of MHC class II proteins used for antigen presentation. This confirmed Pio del Rio Ortega's postulate that microglial cells function similarly to macrophages by performing phagocytosis and antigen presentation. The brain and spinal cord, which make up the CNS, are not usually accessed directly by pathogenic factors in the body's circulation due to a series of endothelial cells known as the blood-brain barrier, or BBB. The BBB prevents most infections from reaching the vulnerable nervous tissue. In the case where infectious agents are directly introduced to the brain or cross the blood-brain barrier, microglial cells must react quickly to decrease inflammation and destroy the infectious agents before they damage the sensitive neural tissue. Due to the unavailability of antibodies from the rest of the body, few antibodies are small enough to cross the blood-brain barrier. Microglia must be able to recognize foreign bodies, swallow them, and act as antigen-presenting cells, activating T cells. Microglial cells are extremely plastic, and undergo a variety of structural changes based on location and system needs. This level of plasticity is required to fulfill the vast variety of functions that microglia perform. The ability to transform distinguishes microglia from macrophages, which must be replaced on a regular basis, and provides them the ability to defend the CNS on extremely short notice without causing immunological disturbance. Microglia adopt a specific form, or phenotype, in response to the local conditions and chemical signals they have detected. Sensum genetics. The microglial sensum is a relatively new biological concept that appears to be playing a large role in neurodevelopment and neurodegeneration. The sensum refers to the unique grouping of protein transcripts used for sensing ligands and microbes. In other words, the sensum represents the genes required for the proteins used to sense molecules within the body. The sensum can be analyzed with a variety of methods including qPCR, RNA-seq, microarray analysis, and direct RNA sequencing. Genes included in the sensum code for receptors and transmembrane proteins on the plasma membrane that are more highly expressed in microglia compared to neurons. It does not include secreted proteins or transmembrane proteins specific to membrane-bound organelles, such as the nucleus, mitochondria, and endoplasmic reticulum. The plurality of identified sensum genes code for pattern recognition receptors. However, there are a large variety of included genes. Microglia share a similar sensum to other macrophages, however they contain 22 unique genes, 16 of which are used for interaction with endogenous ligands. These differences create a unique microglial biomarker that includes over 40 genes including P2Ry12 and hexbook. DAP12 appears to play an important role in sensum-protein interaction, acting as a signaling adapter and a regulatory protein. The regulation of genes within the sensum must be able to change in order to respond to potential harm. Microglia can take on the role of neuroprotection or neurotoxicity in order to face these dangers. For these reasons, it is suspected that the sensum may be playing a role in neurodegeneration. Sensum genes that are upregulated with aging are mostly involved in sensing infectious microbial ligands, while those that are downregulated are mostly involved in sensing endogenous ligand. This analysis suggests a glial-specific regulation favoring neuroprotection in natural neurodegeneration. This is in contrast to the shift towards neurotoxicity seen in neurodegenerative diseases. The